DNA, meaning deoxyribose nucleic acid, is built from monomers called nucleotides. The structure of DNA consists of two strands of nucleotides wound about each other. This forms a double helix. Nucleotides are composed of a ring-shaped sugar called deoxyribose, a phosphate group, and a nitrogenous base. The four types of nitrogenous bases are thymine, adenine, cytosine, and guanine. Thymine and cytosine are single ring structures called pyrimidines. Adenine and guanine are larger double ring structures called purines. Bases are generally referred to by their first letter, so either T, A, C, or G. Hydrogen bonding exists between these nitrogenous bases. Hydrogen bonds are weak chemical bonds that occur between hydrogen atoms and more electronegative atoms like oxygen, nitrogen, and fluorine. The nitrogenous bases of nucleotides form specific complementary base pairs. The purine adenine pairs with the pyrimidine thymine, and the purine guanine pairs with the pyrimidine cytosine. A pairs with T, G pairs with C. So, the bases on the second strand are determined by the sequence of bases on the first strand, because each base must pair up with its complementary base. Additionally, the structure of DNA is antiparallel. As mentioned before, the DNA molecule is composed of two strands held together by hydrogen bonds. A single strand is different at its two ends. One end is called 5' because the strand ends with a phosphate group, and the other is called 3' because it ends with a hydroxyl group. The different strands in the helix run in opposite, antiparallel directions. At each end of the double helix, one strand is 5' and the other is 3'. The antiparallel structure of DNA is important in DNA replication because it replicates the leading strand one way and the lagging strand the other way. During DNA replication, the leading strand is replicated continuously, whereas the lagging strand is replicated in segments known as Okazaki fragments.
There are two types of nucleic acids involved in protein synthesis, DNA and RNA. DNA is located in the nucleus and is composed of deoxyribose sugars. RNA, ribonucleic acid, is composed of the sugar ribose, rather than the deoxyribose, which is what DNA is composed of. DNA and RNA are both composed of four different bases and sugars. The difference between the compositions of DNA and RNA are that RNA contains the nitrogenous base uracil instead of thymine of DNA. Both uracil of RNA and thymine of DNA pair with adenine. The other components of RNA are the same as those for DNA. RNA typically forms a single, sometimes twisted strand, while DNA forms a double helix. DNA consists of a long chain of nucleotides, while RNA consists of a shorter chain of nucleotides. There are three different types of RNA. They each play different roles in the cell and protein synthesis. Starting with transcription, RNA polymerase creates an RNA molecule called messenger RNA from a DNA template strand. mRNA transcribes the genetic code from DNA into a form that can be read and used to make proteins. mRNA also carries genetic information from the nucleus to the cytoplasm of the cell. tRNA, which stands for transfer RNA, is located in the cytoplasm and brings or transfers amino acids to the ribosome that correspond to the three nucleotide codons of the mRNA. The tRNA molecule must become bound to the appropriate amino acid and then recognize the appropriate codon in the mRNA. The amino acids then can be joined together and processed to make the polypeptides that make up proteins. For each mRNA codon, there's a specific tRNA that matches it. rRNA, or ribosomal RNA, is located in the cytoplasm of the cell. In the cytoplasm, rRNA and protein combine to form a nucleoprotein called a ribosome. The ribosome serves as the site and carries the enzymes necessary for protein synthesis. The initial RNA transcripts have regions of non-coding nucleotides that interrupt the nucleotide sequences that actually code for amino acids. These non-coding regions are called introns. Exons are the coding regions of the RNA transcript, the parts of a gene that remain in the mRNA and will be translated. A process called RNA splicing removes the introns and joins the exons together to produce a mRNA molecule with a continuous coding sequence.
The genetic code is universal, meaning that all organisms share the same genetic code book. The genetic information that is stored in mRNA and used to assemble amino acids is read in 3-letter codons. Each codon consists of 3 nitrogenous bases. Each codon represents 1 amino acid out of the 20 that are used in the construction of a polypeptide. There may be multiple codons that code for each amino acid. Codons are deciphered using the mRNA amino acid table. The table is used to decode a sequence of codons as a sequence of amino acids. To read the table, first look for the first base of the codon, find the row on the left labeled with the corresponding letter in the table. Then, look for the second base of the codon. Find the column with the corresponding letter that intersects the row previously selected. Finally, with the selected box, find the codon inside of it and read the amino acid located next to the codon. For example, to determine what amino acid the codon ACG decodes to, you would find A on the left row, C on the top column, and G on the right row. ACG is 309. The ribosome, which coordinates the functioning of mRNA and tRNA, consists of 2 parts or subunits that are each made up of proteins and a large amount of ribosomal RNA. A ribosome has a binding site for mRNA on its small subunit and 2 binding sites for tRNA on its large subunit. The P site, one of the tRNA binding sites, holds the tRNA carrying the growing polypeptide chain. The A site holds a tRNA carrying the next amino acid to be added to the chain. The ribosome connects the newly arrived amino acid to the growing polypeptide chain. The unique structure of tRNA molecules enables them to both become bound to the appropriate amino acid and recognize the appropriate codon in the mRNA. tRNA molecules are about 80 nucleotides in length. One end of a tRNA molecule has a specific triplet of bases called an anticodon. The other end has a site where a particular amino acid can attach. The anticodon is the site of the 3-base sequence that recognizes and matches up with the codon on the mRNA molecule. It must make a perfect complementary match with the codon on the mRNA before the amino acid is released. Once it is released, it is added to the growing polypeptide chain by enzymes.
A mutation is any change in the nucleotide sequence of DNA. There are 3 basic types of mutations: frameshift, substitutions, and inversions. Insertions and deletions can cause frameshift mutations, which is when a base is either added or removed, causing a shift in the reading frame. This results in a completely new sequence of amino acids. A substitution mutation is when one base or nucleotide is replaced with another. A substitution mutation could potentially result in no change to the protein, because in genetic code, several amino acids can be coded from more than one codon. However, there is still the potential that a substitution could drastically change the polypeptide. Generally, an insertion or deletion is more dangerous than the effects of a base substitution. Inversions occur when the specific segment of the chromosome breaks apart, is reversed, and then rejoins to the chromosome. The ending gene is then read at the beginning instead. There is no immediate potential loss of gene material in inversion mutations. However, some of the genes will be in a reversed order. Silent mutations are mutations that are seen to be neutral at the time they occur. They do not result in a change to the amino acid sequence. For example, both AAU and AAC decode to the amino acid asparagine. If a mutation to DNA causes for the mRNA codon to change from AAU to AAC, there would be no change in the resulting protein product.